Okay. Good day, everyone. This is Ma'am Rosc, and once again, we're here to discuss the shoot system. This time, though, we're going to discuss the leaf. Okay. So, to continue with our discussion, we're going to follow the same um, steps in discussing everything. So there would be a general description of the leaf, and then there would be discussion on the external structure, and then the internal structure, and then function and modified leaves. Okay, so to begin with, we simply define the leaf. So we have here the definition, as you can see in your notes. So it is the principal appendage rather borne by the stem. Okay, so the leaf is a flat and an expanded organ, right, most of the time. If it is not modified, this is how leaves look like. No, they are flat and they are expanded. Okay, so the most important function of the leaf would be photosynthesis. It would be absorption or capture of light or light energy for photosynthesis so that they can make their own source of energy. Now, discussions on photosynthesis has already been done, and that should guide you in trying to understand the main function of the leaf. Okay, so during the earliest stage of the leaf, during organ formation, the very first sign, right, of leaf formation would be the formation of what we call the leaf buttress, or what we call the leaf primordia. Okay, so that is the um, origin of leaves. Okay, now when it comes to arrangement, right, leaf arrangement on the stem is what we call phyllotaxy. So there are different types of arrangement of leaves on a stem, right? So arrangement of leaves on a stem is actually highly imperative or significant in terms of avoidance of shadowing. Okay, so imagine if a leaf doesn't have a particular arrangement in the stem, and the leaf are just arranged one on top of the other, then the efficiency when it comes to absorption of light for the main function of the leaf would actually be less. Okay, because one leaf will shadow the other if it is just one on top of the other. So there are different types of arrangement of leaves on the stem. Okay, so there's such an arrangement where we can observe there is only one leaf per node. Okay, this type of arrangement is what we call alternate, right? That is if one leaf per node is just left, right? But if there's one leaf per node, and on each node it is going on different directions such that it is arranged in a spiral manner, then one leaf per node phyllotaxy can then be considered spiral. Okay, and then there's the phyllotaxy, which we know as or observe as two leaves per node. So you will find a left and right pair of leaves, so they are facing each other. Okay, so two leaves per node can either be opposite or it can be decussate. So opposite is when the arrangement is two leaf per node, and then on top of it, the same arrangement or the same orientation of leaves can be found. Now it is disticus or decussate if the second leaf arrangement was like 90 degrees, right, from the one that was below it. So we have photos here to show you how it looks like. Um, However, we proceed to the three or more leaf on one node, and we call that the world phyllotaxy or type of leaf arrangement. Okay, so in this particular photo, you will notice the alternate, leaf arrangement as an example. So you will notice there is one leaf per node, one leaf per node. However, these leaves are actually arranged in such a way that they are in alternate direction, no, per node. Okay, so one goes left, the next node it goes right, the next node it goes left, and so this is alternate. So it becomes spiral if, for example, so not directly from this node, in the next node it's like it goes here, right? Pero it's like 35 degrees, no? Not directly above, no? And adjacent doon sa leaf na nasa baba. Okay, so it goes like the arrangement goes like this. Right, so small angles as it goes up, so that's spiral. Okay, so dun sa next example natin, we have opposite. This one shows, of course, two, no? Two leaves per node. So you have there your right and left leaf. And then another two, another two, another two. This is opposite. Now, this is an example of leaf arrangement with two leaves per node. Okay, so if there's more than one leaf per node, then we already call this type of arrangement world. Now, kanina, doon sa inyong notes, if you notice, mayroon tayong dalawang klase ng two leaf per node. Example, so we have the opposite and the disticus or the decussate. So this is basically how decussate looks like. So you notice that there are two leaves, no, emerging from one node. However, at 90 degrees, the orientation of the next two leaves is actually 90 degrees, no, from the original orientation of the first two leaves. So this is decussate. So this is how decussate leaves looks like. Okay, so these leaves, yung mga mas malaki na yan, nasa mas ilalim yan. Okay, and they are opposite of one another. And then on top of these two would be these smaller leaves, no, arranged at a 90 degrees angle, pero opposite pa din. So you still have two leaves per node, pero right? Hindi siya katulad ng opposite na nasa same angles, no? Yung leaves na kasunod, right? So ito ay medyo parang paikot. So may 90 degrees, 90 degrees again, then 90 degrees again. So this type of two leaves per node phyllotaxy is what we call decussate. Okay. So that's that for phyllotaxy. So we move on to the external structure of the leaf. So there are just very few, parts of the leaf since it is quite well, not simple but small organ compared to the stem or the fruits or the flowers. Okay. So of course, firstly we have the blade. This is the flat portion of the leaf. Okay. Blades of leaves can either be simple blade, right? So if it is just one big blade, right, or a single blade that is connected to the stem, then we call that the simple leaf. So if, for example, this is your stem, so there's petiole, and then you have one blade, so this is called a simple leaf, right? Now, if you have two or more blades, right, in the area where you should be finding the leaf, then you already have what we call the compound leaf. Okay, a compound leaf has different types. Okay, so we have pinnately compound leaf if the leaves are attached to a rachis. So ano yung itsura nito? So for example, this is your stem, right? Then you have a petiole, right? Your petiole extends up to the tip of the leaf. So supposedly here is where you're going to find the blade. Pero sa pinnately compound leaf, instead of finding the blade, you find leaflets, smaller leaf blades. Okay? These smaller leaf blades are connected to the petiole, okay, by a rachis. So there would be these small extensions of the petiole, and then you will find smaller leaves here. Okay. Say for example, okay. Yan. So these are what we call the leaflets. Oops, sorry, naka-eraser tayo. So this is what you call the leaflets, yung mga maliliit na leaf. Okay? So we call them leaflets. Ang pangit ng sulat ni ma'am. Okay? Ulitin natin. We call them the leaflets. Okay? So those are the leaflets. Now this would be your petiole. Okay? And connected to the connecting the leaflets to the petiole will be the rachis. Okay? So ito yung rachis. So when leaves are attached to to a rachis, okay, when it is attached to a rachis, then we have a pinnately compound leaf. Now, some leaves, however, are also compound leaf. However, they radiate from the tip of the petiole. So if, for example, this is your stem and this is your petiole, instead of, or say for example, this is your petiole. So instead of having rachis growing over the petiole like that, okay, the leaflets are actually connected here at the tip of the petiole. So we call these types of leaves palmately compound leaf. Okay. Now, as was mentioned earlier, so you have petiole, okay, and then you have the rachis, and then sometimes, so if this is your stem, and you have a petiole, okay, and then you have a rachis extending out of the petiole, okay, you have a rachis. Sometimes the pinnately compound leaflets are also pinnately compound. So instead of having just one leaf growing out of the rachis, there are several smaller leaves, okay, growing out of this. So these are all still leaflets, but the extension that is connecting these leaflets to the rachis are what we call petioles. So this would be your petiole, this would be your rachis, and these small extensions would be the petioles. Okay? Right. Ay, sorry, that's supposed to be L. Okay? So those are the petioles. Okay, so ayusin natin ng konti yung drawing. So sabihin natin, right? You have the petiole, okay? That's your rachis. So if there is smaller leaflets instead of just one leaflet connected to the rachis, then this is a pinnately compound leaf. Okay? So these small extensions are what we call petioles. Okay? Ito. Yan. Yung mga maliliit na yan. So from petiole, that is what this is trying to say, right? The connection of pinnately compound leaf from petioles would be rachis and then petioles. Okay? Now, in some plants, they don't have petioles. We call them epitiolates. Okay? Epitiolates are normally common in monocot plants, right? And in monocot plants, instead of finding petioles, you will actually find leaf sheath. Okay, and we call these the sheathing base, right? So if this is the stem of a monocot, normally you will find an extension of the leaf that goes like this, right? And this extension of the leaf, okay, normally looks like it is wrapped around the stem. Okay, so this extension of the leaf is what we call leaf sheath. Okay, so leaf sheath instead of petioles, and these types of plants are what we call epitiolate plants. All right. Um, Next we have the venation. So we started from the blade. Okay, so you have the blade, the flat, broad part of the leaf, and then you have simple leaf, and then if it has leaflets, then it's compound leaf. So you have pinnately compound and palmately compound, and then you have the petiole, the part that is connecting the leaf to the stem. Okay, I have photos of this later, so that can also guide you. And now we go to the veins, the ones that are actually found on, well, embedded, right, inside the blade. Okay, so leaf venation could either be parallel or netted, or what we call the reticular venation. Okay, so in terms of parts of the leaf, external structure, the morphology, that is, so this would be the leaf lamina, is also known as the leaf blade. Okay, so that's your leaf blade. The stalk, con um, the stalk that holds the leaf, right, or connecting the leaf to the stem, is of course what we call the petiole. Okay, so this is your stem, that's the node, right? And then we have, of course, the midrib. Later on, we will discuss that when we go to the veins, right? For the internal structure. So you have the midrib is one of the veins. It is the biggest and midmost vein. And then, of course, you have the smaller veins. Okay, so we have two types, as was mentioned earlier. You have parallel and netted, or reticular or reticulate venation. So parallel venation is normally seen in monocots normally, right? And parallel venation is when you will see literally veins that are oriented in a parallel manner. Okay, you will not see veins coming from an angle. Okay, so when you look at monocot plants, you will notice that the veins of their leaves are actually oriented very simply, and it is oriented in just one direction, parallel to one another. Thus the name parallel venation. Now there are two types of netted or reticular venation. If the venation starts from a single point, like you can see here, no, and then from that single point, all the veins radiate, then that is a palmated netted venation. But in most leaves, what you will see would be the pinnately netted venation or net vein leaves, and this is what we normally use when we're drawing leaves. So this is um, the type of netted venation where it comes from one particular axis, and all of the veins are actually stemming from there, unlike palmated where it actually originates from one point, right, at the top of the petiole. Okay, so basically that's that for venation, and that's that for the external structure of the leaf. Quite easy. Right, so for the internal structure of the leaf, we start from the outside as per usual, going in. So the outsidemost part of the anatomy of a leaf will of course begin with the epidermis. So epidermal cells are normally polygonal in shape. Most of the time, they look rectangular or a little squarish. Depends on how big the leaf is, or which part of the leaf you are. Okay, but it is polygonal in shape. Okay, however, there are epidermal cells that are modified. Okay, and these modified epidermal cells basically takes a different or develops a different function. For example, a bulliform cell. Bulliform cells are normally found in monocot, and bulliform cells are normally responsible for rolling of the leaves. Okay. So in monocots, you will notice that when monocot leaves are younger, they are not well spread. No, hindi sila spread up during early stages of their lives. It's like they're rolling, no, dahan-dahan until maturity comes and they are well expanded. Okay, so bulliform cells are the ones responsible for rolling up the leaves. Bulliform cells are larger than most normal epidermal cells, and they are highly vacuolated and they contain water. Okay, so this would be their function. All right. So another thing is they are highly vacuolated. Okay. And yeah, so since they are highly vacuolated, they of course contain a high amount of water. Okay, so aside from bulliform cells, there are also it's there. So aside from bulliform cells, there's also myrosin cells. Myrosin cells basically contain fatty acids. This is highly species specific, and you can find it in almost all of the plants. Okay, so very highly species specific. Okay, so lithocysts, this is quite common. Lithocysts are cells that contains crystals, also known as cystoliths. Okay, we have photos of the bulliform cell and lithocysts later. However, myrosin cells, since they are very um, rare, well, not really rare, pero hindi mo sila makikita in most of the higher plants. There are basically very few photos that you can see of myrosin cells. However, if you see them, since they contain um, fatty acids, you will notice that they look um like bubbles. That's how they look like, no? Pero they don't look like they have empty spaces, but they look like bubbles under the microscope. Okay, so let's go again to lithosis. So lithosis are specialized epidermal cells, or modified epidermal cells rather, that specializes in housing crystals. Okay, so you will normally find cystoliths or crystals in these particular cells. Okay, so also in the epidermis, or in the epidermal area, you will find the stomata. Okay, the stomata consists of two guard cells. Okay, these two guard cells basically regulates the closing and opening of the stomata as an apparatus. Okay, so within the stomata, right? So normally we see stomata looking like this. Okay, so these are stomates, right? If you are looking at it from the top of the cell. Okay, so these two would be the guard cells. Okay, itong dalawa na to, the bean shape. And then the area, okay, let's change the color. This area, yung gitna, right? That would be the stomatal pore. Okay, so this would be the stomatal pore. And then in different types of plants, no, there are different numbers of subsidiary or accessory cells. So, wait, clear natin. Ulitin natin. So, if this is your stomach, okay, write this more than, right? So that's your stomach, or that's the stomata. Okay, this is the, sorry, I'm erasing everything. All right, change color. This would be the stomatal pore. Okay, and then in some leaves, or sorry, in different species, rather, there are different numbers of subsidiary or accessory cells. So subsidiary or accessory cells will normally be found flanking your stomates. Okay. Some of them have two on either each side. Others have four, where you can find two more subsidiary cells along those areas. Okay, so different types of stomates are actually classified according to the number of subsidiary cells that they have. Okay, that would be supplemented in your laboratory. Okay, so um, different species of plants also have different locations where you can find the stomates in their leaves. So if the stomates are found on top of the leaf, right, you call these leaves epistematic leaves. Okay, so on top, so stomates are found on top of the leaves. Those are epistematic leaves. Hypostomatic leaves, of course, you will find the stomates below, no, the leaf, right? So instead of finding them on top, you will find it actually below the leaf. And then antistomatic is are those types of leaves where you can find stomates on both the upper and the lower epidermis. Okay, so right, to be more clear, so epistematic, you will find this in the upper right side of the leaf. Hypostomatic, you will find on the lower side of the leaf. And ampistomatic, you will find it both in upper and lower parts of the leaf. Okay, so those are ways to define or describe your leaves in terms of the area, right, where the stomates are present. Okay, and then of course you have trichomes, and we have different types of trichomes. So trichomes should have been discussed in the tissue, not discussion. Trichomes are outgrowth of the epidermal cells. So they are epidermal cell outgrowth once again, and they have different functions: protection, storage, what else? Uh, area where water is released, and so on and so forth. Secretion, which also equals to protection most of the time. So there are also different types of trichomes. We have simple trichomes, like simple hair-like trichomes. You have branching trichomes. You have glandular trichomes, and so on and so forth. So I will not reiterate on this because I trust that this has already been discussed in the tissues, right, portion of the discussion. Okay, now this would be examples of, right, so here you will see an example of a cross section of the leaf. So you find here the mid vein, which means this is the midrib. No. So ito yung pag hinawakan niyo yung leaf sa ilalim, ito yung mataba, pinakamatabang vein sa gitna. Okay, so ito yun, dyan yung vascular tissue niya. Okay, and then you will notice the polygonal, epidermal cells, which is basically everywhere. Yeah. Okay. So most likely you have in the areas where there's none, most likely what you have is a stomata. Okay. So what I want to show in this particular picture are the bigger epidermal cells. Okay, and these are modified epidermal cells, which we call bulliform cells. So they are the ones responsible for rolling of the cells. Okay, and then on the other side of this figure, you will notice basically the same, right? So, except naka-zoom in lang tayo ng konti. So, hindi nyo na nakikita yung ibang parts of the leaves. So, we are zoomed in the area of the epidermis. Okay? And then, right here, you will notice the lithosis. This is still an epidermal cell. It is not just a space. It is still an epidermal cell, and it contains their cystolith, or yung tinatawag natin na crystals. Okay? And then, notice here, this is actually, clear muna natin, right? Here, in this region, you will actually notice, sorry, in this region, you will notice a stomata. Okay, so this is one of the guard cells. And this area, of course, is the stomatal pore. Okay, yung space sa gitna. So that is how you know that you're actually looking at a stomata in cross-section. Okay, so these are the different types of specialized or modified epidermal cells, and this is basically how a cross-section of a leaf looks like. Okay, so moving on, we now have the mesophyll of the leaf. So kanina nasa epidermis tayo, so now we have the mesophyll. The mesophyll is basically all the space that you find in between the epidermal cells. Okay, so there are two regions of the mesophyll. You have the palisade mesophyll and you have the spongy mesophyll. So starting with the palisade mesophyll, this is an area that is highly photosynthetic. No? You will normally find palisade layers arranged underneath the upper or beneath the upper epidermal layer. Right? There are different types of palisade layers, or rather, there are different types of leaves wherein one can have one palisade layer and the other can have two palisade layers. Okay, so if a leaf only contains one palisade layer, normally you find it beneath the upper epidermis. If it is by palisade layer or bilayer of palisade, then you will notice it is arranged beneath the upper epidermis and before the lower epidermis. And it sandwiches the second kind of mesophyll, or the second area of the mesophyll, which is the spongy mesophyll. Now, it is it is said to be a highly photosynthetic layer because, and this you will notice later doon sa ating figure, there are cells, right, that contains chloroplast, that of course contains chlorophyll, and these cells are arranged in a highly compacted manner. Okay, and because of the abundance of chloroplast, right, in this region, this tissue, the palisade mesophyll, is normally called chlorenchyma. Chlorenchyma. That's because they are basically parenchyma cells that contains chlorophyll. So you call the tissue a chlorenchyma tissue. And then we have the spongy mesophyll, and the spongy mesophyll is always found beneath the palisade layer. So later on, dun sa ating figure, you will notice kung saan sila nakalagay. Now, in contrast to the palisade mesophyll, which is highly compact in nature, the cells of the spongy mesophyll is highly loose. And despite the fact that the cells of the spongy mesophyll also contains chloroplast, that of course contains chlorophyll, photosynthesis is not the main function of the tissue, right? Or this area, because the highly large space, no, air spaces or irregularly arranged cells actually provides for big air spaces in the area, and so the main function of this particular tissue now becomes gas exchange. Okay, so it is not wrong that cells of the spongy mesophyll can photosynthesize because they still contain chloroplast and they contain chlorophyll. But because of the loose arrangement of the cells, this now provides a lot of air spaces that can uh, provide a region that is highly efficient for gas exchange. Okay, now, since this area contains a lot of loosely arranged cells, right, the whole tissue is then termed parenchyma. Most parenchyma are responsible for gas exchange. So these are types of parenchyma cell, and they are specialized in gas exchange. Okay. All right. So discuss muna natin yung vascular tissue before we go into the figure, because you can also see the veins in the figure. Now, the vascular tissue are basically the veins of the leaves, right? So you will notice these veins are arranged in such a manner that there is one big mid vein, right? And this is called mid vein because it is in the middle. Okay, so mid vein, and then from a mid vein normally comes out lateral vein. So di ba pag nagdo-drawing tayo normally pag nagdo-drawing tayo normally ng leaves, ang ginagawa natin, hide floating. Okay. Pag nagdo-drawing tayo normally ng leaves, okay, what we normally do is, so we draw the lamina or the blade, and then we draw the midrib, right? Ito yung nakikita mismo sa gitna. And then from there, we normally draw veins coming out of the midrib, and these are the lateral veins, okay? And then, from the lateral veins, there are veins that are specifically found dito sa dulo-dulo, right? Sa pinakadulo, right, ng leaves. And we call those the ultimate veins, okay? So, those are the different veins, and they are actually named depende sa kung gaano sila o kung saan sila nakikita, okay? Now, mutation, and this you will see in one of the functions of the leaves later. Mutation is actually, oh, I don't have a video, sorry. Show video panel. There. Mutation is actually one of the ways of elimination of water, or plants eliminating water. No? So, this normally happens at night. It is different from transpiration. Transpiration is brought about by evaporation of water, and this normally happens through the stomata. Mutation happens in special cells, which we call the hydatodes, which you will notice later, but I will write na din. So there are special secretory cells known as hydatodes, and hydatodes are responsible for elimination of water during nighttime. This happens normally because the soil contains a lot of water, and so the roots are absorbing a lot of water, and the taught water column in the xylem area is full. So without the sun or any source of heat that allows for evaporation of water, the taught water column will stop, right? And the roots would stop absorbing if hindi lumalabas yung water. And so parang napupuno, no, yung xylem mo, and for the plant to be able to absorb water continuously, what happens is nagkakaroon ng secretion of water through the hydatodes. Now, the ultimate vein is the one that is connected to the hydatodes, and siya yung immediate na vein na nakakonect doon sa secretory cells na ito, and this is where water is actually expelled. Okay? Kaya kapag umaga, 'di ba, makikita niyo lalo na yung mga grass, makikita niyo may mga basa sila, 'di ba? Pag sa morning naglakad kayo through the grass, basa sila, and then you will notice the dew, no, na tinatawag natin. So that's because of the process known as mutation happening at night. Okay. So aside from the veins, you will notice that the veins is actually surrounded by a tissue, no? And this tissue we call bundle sheath. Okay, so the bundle sheath is a single layer of parenchyma cells, and they surround small veins. And the reason why there is bundle sheath is because the leaf is basically a very how do you say this, delicate organ, no? And the veins, right, in the leaf blades are actually highly delicate. So they are surrounding the veins to provide support or protection, right? Because this tissue, yung mga veins na ito, maliliit sila and they're highly delicate. So they need extra support. And that's what bundle sheets are for, okay? And so we now have figure that is showing us basically the mesophyll region. Okay, so here you will notice the upper epidermis. Okay, and right after the upper epidermal layer, you will notice this layer, which is a compact layer of cells. How do you know that it is highly compact? You will notice na it is dikit-dikit or it is oriented one after the other in comparison to this area, which is the spongy mesophyll area. The spongy mesophyll area provides for more, no, air spaces because of the um, loose arrangement of cells in there. Okay, so notice na in the cells of the spongy mesophyll, there are also chloroplasts. Okay, so these chloroplasts, just like the chloroplasts that are found here in the palisade layer, they also function for photosynthesis. Pero gaya ng sinabi ko kanina, because of the loose arrangement of the cells in the spongy mesophyll region, that provides basically for a lot of air spaces, then the main function of the spongy mesophyll, of course, now becomes for gas exchange instead of it prioritizing or um, having a main function being photosynthesis. Okay? So, mayroong mga plants, however, na makikita nyo, I think you're going to have samples of this in your lab, hopefully you do, wherein after the upper epidermal layer, you have the palisade layer, okay? And then a spongy mesophyll, and then you will have another palisade layer. So these are the ones that has bilayer of palisade cells. Okay? So yung most of the plants only have one layer, and when a plant only has one layer of palisade tissue, normally it is found right beneath the upper epidermis. Okay, that is something that you have to take note. And then, of course, here you will also notice the vein. Okay, oh, we're on eraser mode. So here, that's the vein, right? So you will notice your xylem and then your phloem. So this is your phloem, this is your xylem, and that would be the bundle sheath cell. Okay, so basically that's how it is. All right. So kung paano niyo nakikita yung yung vascular bundle system at saka, so basically that's how it looks like din sa leaves, no? Wala nga lang inside. So xylem is on top and then phloem is below. Right? Kasi hindi cylindrical yung ating leaf. Okay. All right. So basically, that's that for the internal structure of the leaf. So moving on, we're now down to the function of the leaf. And of course, the main function of the leaf is photosynthesis. Okay. So we're not going to discuss photosynthesis as a process because this has already been discussed. right? So that discussion should guide you, no, once again, into trying to understand this. So we're just leaving you a few notes on photosynthesis. So leaf is the major site of photosynthesis in plants. Basically, when it comes to cells, any cells that contains chloroplast, therefore any cells that is green, can actually photosynthesize. But the leaf is the one that is most efficient when it comes to capture or absorption of light energy. Therefore, it is the major site of photosynthesis. Okay, so since chloroplasts are the site of photosynthesis and they are found mostly in the leaf and highly abundant in the palisade layer, this also contributes to the efficiency of the leaf being the major site of photosynthesis. Okay, and then of course we have gas exchange. A few things that you have to remember about gas exchange. Why is gas exchange necessary? Number one, when leaf are photosynthesizing, they are actually emitting oxygen. So a way of releasing that oxygen should be possible. And this is made possible by the air pockets, right? And of course, the stomach. Second, during photosynthesis, carbon dioxide is needed para makaproduce ng sugar, yung mga leaves, so, or yung mga plants. So, an intake of carbon dioxide should also be facilitated. And the air spaces and of course the stomates also helps provide for this particular function. Okay. Aside from that, during cellular respiration, when cells are utilizing organic compounds so that they can produce energy or ATP, what they normally do is they utilize naman oxygen and they release carbon dioxide. So, kailangan ng oxygen na pumasok sa loob ng body ng halaman. Okay, and this is also done through the leaves, okay, through the stomates. Okay. Now, the gas exchange, the whole gas exchange idea is facilitated by the stomates. So the gas exchange, as is noted here, occurs in the stomach. Now, if the stomates, right, facilitates for this, of course, naturally, there's opening of the stomates and closing of the stomates. Okay. So, na remember nyo, we have the guard cells. Okay? Yung guard cells, yung isang wall ng guard cell, the one that is inside, it is actually thicker than the one that is outside. Okay? So, when the guard cells are flaccid, basically the stomates are closed. But when the guard cells is full of water, right? Parang balloon. Yan, no? So, nag-e-extend siya. And because mas makapal yung wall sa loob, okay, gawin natin mas makapal. Okay. Yan. Mas makapal yung walls inside, sorry, undo, inside the guard cell. Kapag na-fill up ng water yung guard cells, ang nangyayari, hinahatak niya ngayon yung thicker na wall. So what happens is, nag-e-extend or nag-distend. Yan, nag-e-expand yung guard cells, right? And then nahahatak niya ngayon yung walls, right? And na-open ngayon yung stomatal pore. Okay, so paano na-open yung stomatal pore? As is written there, opening of the stomata is caused by an uptake of potassium ions. So potassium ions will enter, right, the guard cells, right? And with the help of blue light, okay, so ito na-detect it, of course, ng halaman. With the help of blue light, right, more potassium, right, will be more potassium influx channels or protein channels will be open. and that will help in what do you call this, movement of potassium. And blue light also, right, is perceived by this photoreceptor, which we call phototropin. So when there's blue light, phototropin basically activates the proton pump, right? This proton pump is ATP driven. So gumagamit siya ng ATP, right? And so ang mangyayari, bababa ngayon yung pH inside the guard cell, and this will open more yan. Open more influx channel for the potassium. And dahil dyan, papasok ngayon yung water. Okay. Now, when it comes to closing of the stomates, right, abscisic acid is actually responsible for this. So abscisic acid will bind on certain proteins, and then this will cause the pH of inside the cell to increase. The pH increasing inside of the cell will now stimulate opening of efflux channels. So pag sinabi niyong efflux, it means palabas. No? These channels are the ones causing exit. Right? So yung potassium ions dito, pag sinabi mong uptake, ang ino-open niyan na proteins ay influx proteins. Okay? Efflux kapag palabas, influx kapag paloob. Okay? So the pH increases, unlike dun sa opening where it decreases, and it stimulates the loss of chloride and other organic ions. And the loss of chloride, right, facilitates for transfer of calcium from the vacuole into the cytosol. So nagkakaroon ngayon ng imbalance of charges. The chloride is negative. Calcium are positive. So mas nagiging positive yung loob, negative yung labas ng environment no stomates, and that difference in electrochemical gradient will now cause more calcium to be released, right? And this will cause more chloride to be released, and then eventually potassium are also released because of the difference in the electrochemical gradient. Okay, the electrochemical gradient nagiging imbalance kasi lumalabas yung mga anion. So napupuno ng cation in the form of calcium, and then dahil nagkakaroon ng electrochemical imbalance, lumalabas din, bakit yan Ca? Lumalabas din yung potassium. Okay. Dapat Ca natin. Yan. Lumalabas din yung potassium. So, lalabas yung potassium, lalabas yung calcium. Ngayon, mas konti na yung solutes, right? Inside the stomates. And that will now cause the closing of the stomata. Okay. So, gas exchange now happens with the help of all these opening and closing of the stomates. Siyempre, pag close yung stomates, walang gas exchange. Pag open yung stomates, may gas exchange. Okay. And normally, photosynthesis starts to happen in at daytime, right? And this is also when CO2, carbon dioxide, is taken in by the plant. Kaya yung opening ng stomates normally also requires blue light. Okay. Now, aside from gas exchange, another function of the leaf would be transpiration. Transpiration basically is the movement of water driven by evaporation of water on top of the plant. So normally this is happening sa leaves, and dahil sila yung exposed sa light, so transpiration is happening there also through the stomates. Okay? And then of course, mutation, na-discuss ko na ito kanina, takes place in special secretory structure known as the hydatodes. And these hydatodes are actually found at the tip and margin of the leaves connected to the ultimate vein, and mutation is a process of expelling water during nighttime. Okay, and it is happening because normally the soil contains high amount of water, and the roots, no, has to continuously absorb water, and if it doesn't expel water sa leaf area niya, then the whole absorption will cease. So kung halimbawa nakatanim yung halaman sa area kung saan mababa ang water content ng soil, then you will not notice mutation. Okay, so nangyayari lang yun kung abundant yung water doon sa soil. Okay, and then leaf abscission. Of course, dahil ito ay leaf abscission, ito ay function ng leaf. So, anong nangyayari during leaf abscission? Leaf abscission basically takes place because of aging, senescence, you know. So, senescence basically means nag-age na yung plant. Kapag nasa tamang age na yung plant, it is old enough, right? It has to get rid of some of the leaves so that the new ones that are growing will be given more nutrients and will be provided with more energy that it needs. Okay. So, pag sinabi nating abscission, as is written here, it is the separation of the leaf from the main stem without causing injury to the stem. Okay, so the abscission of leaves is actually facilitated by the hormone known as ethylene. Okay, so some of you would have answered abscisic acid, but it's not abscisic acid. It's the main hormone for leaf abscission is actually ethylene. Okay, in any case, sa stem, right? Kapag makita nyo to under the microscope. In a stem where a petiole is connected, see for example, itong area na to, no, mayroong area dyan, doon sa junction niya, that we call the abscission zone. Okay? The abscission zone is the specific zone where a leaf will abscise during proper leaf abscission, ha? Hindi yung pagpinitas nyo yung mga leaf, right? During proper abscission phase, and kapag natanggal dun yung leaf, right? Sinisil yung area na yon, and that is how injury is avoided. Okay? Now, in this abscission zone, mayroon tayong tinatawag na separation layer. The separation layer contains delicate layer of tissue. Nakaikot yan, paikot. No? Makikita nyo under the microscope. Okay. Now, the main target of certain enzymes during abscission would be the cell walls of the separation layer. Okay? So, dun sa cell, with the help of ethylene, right, certain hydrolases would be produced. Right? One of these would be calcium pectate. Calcium pectate will then turn into pectic acid. That turns into pectin. Pectin basically makes the cell wall of the separation layer weak. Right? So, if abscission time na, magpo-produce ng ethylene yung leaves, ipapadala niya dito, yan, towards that area, right? And the cells in the separation layer will start to produce calcium pectate, turn it into pectic acid, then pectin, and then the cell walls of the cells in the separation layer will start to weaken. Once that happens, nagkakaroon ngayon ng weak attachment yung leaf doon sa stem. And as the cell wall weaken, the cells die. So once the cells of the separation layer die, that leaf will abscise from the stem, right? And that's how leaf abscission basically happens. Okay, so we're done with the function of the leaves. So moving on, we have specialized leaves, starting with succulent leaves. Oh, yung mga mahilig mag-garden dyan, no? Or yung mga mami nyo, so you're very familiar with succulents. Succulent leaves are thick-lipped. leaves. Na they store a lot of water, okay? And this is due to their parenchyma being highly vacuolated, and that's the reason why they can store a lot of water. They also have very thick epidermis and very thick cuticle to prevent water loss. Why? Because most succulent leaves are actually found in very arid areas, no? Like the desert. Yan. So, sa mga maiinit. So, naging part ng aesthetic na lang ng garden or ng house yung mga succulents these days. That's why you can see a lot of them around. But in nature, you will normally found succulents in area where it is really hot and evaporation of water is high. So they have very thick epidermis and they have very thick cuticle in their epidermis to prevent water loss. Okay, and then after that we have spines. Spines are actually modified leaves. No? So they are actually modified leaves. Okay, these are leaves that are pointed, and they are normally found in, well, a lot of plants, right? Pero parang sabi nga dito, like those found in desert plants, sa mga cacti, but they have spines. It's basically the same. So you can see in the figure below how spines look like. You will find this in calamansi. You will find this in, well, sa rose it's thorns. No, basically they are not spine. So spines are highly pointed. Mahaba sila. Okay? So they are used as defense structure. Their mesophyll is greatly reduced, as you can see, kasi medyo woody na din sila in nature. And remaining cells develop thick walls that becomes impregnated with lignin. Lignin makes any structure highly sturdy. No? Kaya parang matigas sila. It's highly because most of the cells of spines are highly lignified. So this is another example of specialized leaf. And then, of course, we have bud scales. Bud scales are actually specialized leaves. They are small leaves, and normally their main function is to protect delicate meristematic tissues of buds. Kaya nga sila tinawag na bud scales. So their main aim is to protect the growing structure, right? Which we call the bud. Okay, so cells, right, secrete substances known as collitor, and this collitor is glue-like. It keeps the leaves in close approximation of one another. Kaya pag nakita kayo ng bud, diba, at mayroon siyang bud scales, diba, hindi yan naghiwa-hiwalay agad, no? Talagang unless you insert force, right? So, dun lang sila maghiwa-walay. Pero kahit tanggalin mo sila from the stem itself, the bud scale remains intact. And this is because of the collitor. Okay. So, number 4, we have insect traps. Yeah. So, this is one example. This is a Venus flytrap. Okay, so insect traps are basically what their name suggests. They trap insects. They are carnivorous, or more specifically, they are insectivorous plants. Some of them will secrete sugary fluid, which we call nectar. No, and this nectar is the one that attracts insects. Yeah. So when it attracts, insects normally can detect this and they get attracted, and that's how they actually land on the leaves. No, some, however, secrete volatile substances through what we call osmophores, and this also provides for attraction of insects. So when an insect is caught, no, dito sa gitna nila. Asa na ito? Diyan, inside. Diyan, sa loob na yan. So, pag na-trap yung insect dyan, the epidermis, no, the glandular trichomes in the epidermal layer will start secreting proteolytic enzymes, and they slowly digest their victim. And then they absorb, no, whatever is left of the victim, and that's how they actually eat them. So kaya sila tinawag na insectivorous. Okay. And then last but not the least, we have tendrils. This one, nilagay ko talaga sa huli kasi baka malito kayo. Kasi baka sabihin, ma'am, akala ko ba ang tendrils ay specialized stem? No. There are tendrils that are actually specialized stem, and there are tendrils that are actually specialized leaves. Normally, medyo mahirap siyang makita, pero a more slender, mas manipis, right? And mas fragile yung mga tendrils that are actually modified leaves. Just like tendrils that are modified stem, pareho lang yung kanilang function, and that is to provide support. Okay? An example of a plant na ang tendrils niya ay galing sa stem would be grapes. An example of a plant na ang tendrils niya ay modified leaves would be cucumber. Yan, or ampalaya. Yan, most of the tendrils that they have are actually modified leaves. So, wag malito, may mga tendrils na modified leaves, may mga tendrils that are actually modified stem. It is highly species specific. So, kailangan nyo lang alamin kung yung tendrils ng isang halaman ay from the leaves ba, or modified leaves ba, or modified stem. Okay, so just like what I said, very short lang yung discussion natin for the leaves. And that basically ends the discussion for this part of the shoot system. So I hope you guys learned something, if not a lot. And if you have any questions, just like usual, you just ask your professors. Okay, so with that, thank you very much for listening.